And we are live. Hello everyone, we are live now. Shreya?
Yes.
Yeah, we are already live.
Okay, so let us start. Good evening, everyone, and welcome you all to the sixth episode of the exclusive lecture series of CAPSS called Nature as We Unfold It. This time, we have a really interesting and very important topic, which is vaccine development. We have with us today two researcher friends of ours from All India Institute of Medical Science at New Delhi, and they would be talking about the various stages of vaccine development, the safety concerns, and timelines. They will also cover on the vaccine candidates those are being developed for COVID-19. So let me first introduce our speakers, Harun Rashid and Aphrodite Chakraborty. Harun Rashid is currently pursuing PhD in the Department of Pharmacology at AIIMS. He does his research in neuropharmacology. And Aphrodite is a senior research fellow at the Department of Neurology at AIIMS. And her current project is to investigate in pharmacin resistant epilepsy, and also she is involved in studying the role of genes in complex human neurological disorders and the use of genetic engineering in regenerative medicine. Now, before we start, there are some requests to the audience. Please keep your microphone off while the talk is going on. And if you have a question, kindly raise your hand and we will give you opportunity to speak. For the public information, I would like to remind you once again that this talk will be live streamed on YouTube and your cooperation will be highly appreciated. Now I request the speakers to kindly proceed.
Thank you, Shreya, for the introduction. Shall we start the presentation
Yes,
now?
yes, sure. You may share your screen.
Okay. Is it visible now?
Yes, yes. It's actually, yeah, yeah. Now it's visible. Perfect.
Okay. So, good evening, everyone. Shreya has already introduced us, and for the purpose of this presentation, I should mention that the molecular part and the immunology part of this presentation will be explained by me, and the clinical research part will be explained by Harun
Introduce
Rashid.
our speakers.
So
Harun Rashid
let's start with the
in
presentation now.
neuropharmacology.
You see, this is the complete overview of the presentation that
Raise your hand
both
and
will
we
be
will give you
presenting.
opportunity to
The
speak.
first thing that we will be
For the
discussing will be
public
immunity.
information, I would
Then we will
like to
discuss about
remind you
what a
once
vaccine
again
is,
that this talk
the
will
difference
be live
between
streamed.
vaccines and drugs,
Share
the
your
types
screen.
of vaccines, compositions of
Is it
vaccines,
visible now?
risks that are associated with vaccines,
Yes, yes.
and
It's
finally the
actually,
protocol of
yeah, yeah.
vaccine
Now
development,
it's
which will
visible.
be divided
Perfect.
into preclinical and
So,
clinical
good evening
investigations.
everyone.
So the
Shreya
preclinical
has
part
already
will be
introduced us,
explained
and
briefly
for
by
the
me, and the
purpose of this
clinical part will be
presentation, I
explained
should
by Harun.
mention that
And
the
although this was
molecular
not
part and the
exactly in the scope
immunology
of our
part of this
presentation initially,
presentation will be explained
we
by
have
me,
decided
and the
that in the
clinical
face of
research
a
part will
raging
be explained
pandemic
by
at the
Dr.
moment,
Introduce
we
our
will be giving a
speakers.
very, very brief
So,
overview
Harun
about
Rashid, start
COVID-19
with the presentation
before
of
moving on to
neuropharmacology.
a brief
You see, this
description
is the
about the
complete
vaccine
overview of
candidates
the
that are
presentation
being
that I would
tested right
like
now.
to give you opportunity to
So
speak.
let me begin.
For the public
The
information,
first
I
thing
would
that we
like to
need to
remind
talk
you once
about
again
is
that
what
this talk
immunity
will
is.
be live
It
vaccines
is
and drugs.
basically
Share
nothing
your
but a
screen.
body's response to pathogenic infection. Now, what a
Yes,
pathogen
yes,
basically
it's
means is something,
actually,
some
yeah, yeah.
external
Now it's visible.
microorganism
Divided
that
into
comes
clinical,
inside
preclinical.
our
So
bodies
let
and
me give
induces
you
some kind of a
clinical
disease or
part
a
will be
disorder in
introduced
our body.
and for the
So, our
purpose of
body
this
as such has
presentation, I
got
should mention
several
that
defense mechanisms to fight against microorganisms that invade our body. Among those defense responses, the first thing that has not been shown here is the physical barrier. which is nothing but our skin, our mucous membrane, etc. Mucus is nothing but the lining of our internal skin. Okay, so the first time a microorganism comes and we get exposed to it, our skin basically stops it. When our skin cannot stop it, it enters some of our body cavities. Let's say it enters our nose. There is a particular system which is known as a mucus, which tries to trap those microorganisms, or there are fine hairs inside our nose which try to expel those microorganisms. Now, in case our physical barriers don't work, then there are two kinds of immune responses which are inside our body. The first one is innate, the second one is adaptive or acquired. What innate immunity is, that it is present inside us right from birth. Adaptive or acquired immunity comes to our body eventually as we keep growing up, and as pathogens keep invading our body starts to recognize particular pathogens and start developing certain immune responses, which is the adaptive immune response. Now, so in summary, the immune response is basically present from birth, and it is a very quick response. It is the first response that happens inside our body once there is a microorganism that infects. And it is also very non-specific. It doesn't care whether there's a bacteria or a virus. It just keeps attacking. Like there are macrophages, these cells which I'm going to name, there are macrophages, there are other types of cells which basically eat up those pathogens. Once that has happened after a pathogenic infection, the second response, which is adaptive response, starts getting activated, which is about 4 or 5 days after the initial infection. What happens is in adaptive response is that it is secondary. It occurs after 4 to 5 days, and in this case, it is very specific. So, for a particular kind of, it's not just about a bacteria or a virus, for a particular kind of virus, for a particular strain of that virus, there is a particular cell line, a line of cells which is basically involved in the adaptive immune response. In immune response of the adaptive kind, there are antibodies which we have all heard about during the current pandemic. There are also some things called as T cells, which basically entire adaptive immune system. So antibodies are released into our body fluids, and T cells basically keep like in which our adaptive immune system responds. Now, why I'm talking about this is because in the case of vaccine response, these things are going to be very important. So, in case of adaptive and innate immune response also, there are two kinds of immune response, which is one is active, in which your own body is producing the immune cells. And then there is a passive kind of immunity in which you are already given ready-made immune cells. So what happens in case of vaccine is that we give a biological preparation which provides an active acquired immunity. Active acquired immunity as in the sense your body is itself producing the immune cells, and this is an acquired or active kind of immune response, so it is specific to the pathogen which is infecting you. The first vaccine was discovered by Edward Jenner. So this is a story, I don't know how many of you have heard about this. This is a very common story that is taught in basic biology classes, that Edward Jenner observed that farmers who milk cows seem to be resistant to smallpox. So he hypothesized that it was because those farmers had been exposed to similar kind of pathogen which infects cows, basically known as cowpox. Cowpox does not really cause a very high immune response or an adverse effect in human beings, but regardless, because of their close contact with the cows, humans were exposed to cowpox viruses. So their body recognized those viruses. The next time when the body comes across the smallpox virus, which is very similar to the cowpox virus, not the same of course, but it is very similar to the cowpox vaccine. So the body already knows that I have been exposed to a similar kind of a pathogen beforehand, and the body protects you from the smallpox pathogen. Smallpox used to be one of the worst kind of infectious disease back once upon a time, but because of the advent of this vaccine and the vaccination procedure, right now smallpox is all but a forgotten thread in our world. Now, coming to the modern world, there are several different kinds of vaccines that are used, but before we go to that part, we need to understand especially in current context, vaccine and drugs are not safe. What a vaccine basically does is it prepares your body to fight against some kind of an incoming infection. It is a preparatory kind of a mechanism. What a drug basically does is it treats your body after the infection has happened, after you have developed the disease. So when we talk about in the current times, when we talk about vaccines, people tend to think that a vaccine is going to cure our pandemic. That's not what's going to happen. What's going to happen is that our bodies are going to get trained to fight against the virus. First, there's a chance that some of us may still fall ill, even after vaccination, as it happens in case of influenza, but we will have a better fighting chance. But it is not a drug. It cannot help a person who has already started an infection process with COVID-19. So going to the next part that I mentioned, the types of vaccines. This is important because in general day-to-day life right now, in the midst of a pandemic, I don't know how many of you go ahead and read about these things, but there are several vaccines which are under development,
Running?
and some of them belong to these different categories of vaccines. So in brief, the different types of vaccines are live attenuated. Attenuated or live is basically you have a virus or you have another pathogen. So before going into this, I'm sorry, I'm just going to interrupt my own self. Before going into this, what I need to explain very clearly is that a main component of the vaccine will be something which is either a part of the infecting pathogen or it will be something resembling the infecting pathogen. So in case of live attenuated vaccines, what we do is that we take a pathogen and we disable the viral property. How we do this is by, like, in labs, we grow viruses, we grow bacteria. So when we do that growing process, we change some kind of experimental conditions because of which the pathogenicity, the capacity of that pathogen to actually infect our bodies reduces. It undergoes certain genetic changes because of which the pathogenicity or the virulence decreases. That is the whole concept of live attenuated vaccines. So the pathogen is still alive, but its capacity to infect has decreased, or to elicit an immune response in us has decreased. Now this kind of vaccine is very much preferred because it is very durable and very few booster shots, very few repeated shots are actually required. So some of the most common live attenuated vaccines that are common currently in use are, we must have all heard of this. This is called BCG vaccine, which is against tuberculosis. There's also an MMR vaccine, which is mumps, measles, and rubella. So these are live attenuated vaccines. Now then there is an inactivated vaccine in which the pathogen is basically killed, either using heat or chemicals or radiation, some form of it. So commonly used inactivated vaccines are polio vaccine, rabies vaccine, influenza shots. The next kind of vaccine is subunit vaccines, in which just a part of the pathogen is actually used. Just like, let's say it would be a part of the protein inside the virus or the bacteria, or some other part will be used, a fragment will be used. So commonly used subunit vaccines are hepatitis B, in which the surface protein of the virus is used, and also human papilloma virus, which is commonly transmitted through venereal mode. Then there is toxoid vaccines. We have all heard about tetanus, anti-tetanus injections, which is a type of toxoid vaccine in which toxic compounds are used, not a part of the pathogen, but the toxic compounds themselves are used as vaccine. The next one is conjugate vaccine, in which one kind of, like, the best analogy would be we take a coat from someone, a bacteria, let's say, and we take the body of some other pathogen, the pathogen of interest. So the coat comes from somewhere else and the pathogen comes from somewhere else, and that is called a conjugate vaccine. Okay, one of the most common conjugate vaccine is again for influenza of type B. The next two candidates are DNA vaccines and recombinant vectors. They are not really very commonly used. They are still experimental vaccines, and some of our most interesting COVID-19 vaccine candidates are also in this group. So coming to DNA vaccine, or there is also an RNA vaccine. So both of these are nothing but part of the genome. So what we do is that we take a virus, let's say, for the sake of this conversation, there will be a virus and we will be extracting the genome of that virus, the DNA or the RNA, and then we will be injecting it into the human being in order to, like, human being themselves have a genome. We want to insert the viral genome into the human genome in order to elicit a immune response. So now the thing is, DNA vaccines and RNA vaccines are, like, if they are successful, they are very good candidates as vaccines because their adverse effects are quite low as compared to the other kinds of vaccines, and they are also very cost effective, and they are also very they act for a very long time. But most of the DNA and RNA vaccines are still undergoing clinical trials, not just for COVID-19, but for others as well. Apart from that, there are recombinant vector vaccines, which is nothing but, you take the part of a pathogen and you put it inside another. Let's talk about COVID-19. You take a part of the COVID-19 genome and you put it into some other virus. So vector is nothing but a carrier. You are using the fragment of your pathogen of interest and you're putting it into a carrier. You can either put it into a virus itself, another kind of a virus, or you can put it into something called nanoparticles. Pretty sure you must have all heard about it. So this was all about the types of vaccines. Coming to the compositions of vaccines in very brief, not all of these components are present in all vaccines. The main component is the active vaccine component, which is the pathogen or the toxic material which is responsible for eliciting our immune response. Other than that, there are certain things called adjuvants. Adjuvants basically help in increasing the immune response. The efficacy of the pathogenic component that we are using as a vaccine component, adjuvants basically add as aid the whole process. So, for example, in the tetanus vaccine shots, alum is commonly used as an adjuvant in order to, make the body respond faster and better. Our vaccine components may as well get contaminated by something else, some bacteria, some fungus, etc. So, in order to preserve our vaccines, we use preservatives or we use excipients such as stabilizers, antibiotics, etc. The last part is the vector that I was talking about. Vector is, let's say you are traveling in a car, so the car is the vector and you are the component, you are the vaccine component. So for the purpose of making vaccines and delivering vaccines, we use different kinds of vectors, and the most common vector that is in limelight these days is viral vector. I will be talking more about this later on. Okay, why later on? Right now, the adenovirus thing that you may see in this slide, that is the I don't know how many of you have heard about the Oxford trials, but the Oxford vaccine is using a very popular kind of adenovirus known as adenovirus type 5. So that is basically a carrier for our candidate SARS-CoV virus pathogenic component. Before coming to the vaccine development process, this is the last slide. This acts as a kind of a warning that for lay people it is very difficult to understand why it takes so much time to develop a vaccine. So there are certain kinds of risks that are associated with vaccines, but even before coming to the risks, sometimes what happens is that vaccines don't really work the way they're supposed to. This may happen because the attenuation of vaccines may not have been done in a proper way. And sometimes the vaccines that have been inactivated actually regain their the pathogenic capacity. Like before vaccines, we have gone through all the procedures, but we might have not taken enough time. And somehow the removal of the components that made the virus dangerous to us were not done in a permanent way. And because of our bad luck or something else, something that went wrong experimentally, sometimes the vaccines can actually revert back to the dangerous form. Apart from that, when you don't test a vaccine properly, there are also host-related failures. So in that case, what happens is that the immune system does not respond in the way it is supposed to. There are human beings are all different. They have different genetic makeup. They have different immune status. Like somebody might have very weak immune system. Somebody might have a very overactive immune system. So in that case, what happens is that if you deliver a very quickly developed vaccine to them, and let's say you tested on a limited number of individuals and you administer it to a larger population, what may happen is that sometimes there are adverse effects, unintended immune response, and sometimes, like, the worst case scenarios are that either the body does not even respond to the vaccines. So in that case, you get infected by the pathogen anyway. But then there is a worse form where your body starts overexpressing its immune response and you develop autoimmunity. Autoimmunity is a condition in which your body's immune system will start killing your own cells, own healthy cells. So there are certain autoimmune diseases in which a person even loses their entire mental faculty, their control over their bodies, and the entire physiological setup goes wrong because the body's immune system starts attacking your own body. So this is a very, these risks are very real when you don't go through the entire proper vaccine development protocol. Even after a proper development protocol, sometimes because these are biological organisms we are working with, they do have some degree of unpredictability. Anyhow, apart from the unintended immune responses, there are certain very common adverse effects which are not that serious, which are like low-grade fevers or local pain and swelling, nausea, etc. Sometimes there are rare adverse effects such as seizures, coma, certain kind of neurological disorder known as Guillain-Barré syndrome, pneumonia, brain swelling, etc. So that is the entire crux of the thing, that one, unless we go through the proper vaccine development protocol, vaccines may sound like very good candidates to save us against some kind of pathogens, but the vaccines themselves may pose very, very great risk to us. Moving on to the protocol of vaccine development, you see the the slide is divided into five sections. So the first section is the exploratory or the preclinical part of the vaccine development. In that particular part of our vaccine development protocol, what we do is that the first step is in silico development. In silico means nothing else happens. We just check our we use computers, we use softwares, and we predict whether a certain molecule will actually interact with our molecule of interest. So let's say that we have a particular candidate molecule, and we want to check whether or not it interacts with a particular type of receptor in our body. So the first thing that we do is that we check it via softwares. Once we are done with the software part, the in silico part, we take it on to the next level, which is testing in cellular setup. For this, we have laboratory. In labs, we do the cell cultures, and in cell cultures, we check different, like after administering those molecules, after injecting those molecules inside the cell, or we try to check that whether in the presence of those molecules, the cell is behaving in the way we want them to behave or not. So this is basically the cell culture part, the in vitro part. After this, we go on to test them among animal models. Like we use rodents, we use primates, etc. And the entire sum of this, starting from the in silico, the computer-based, program-based development, then coming to the in vitro, the cellular part of the development of the vaccine, and then going on to test them in animal models such as rodents or primates or some other animals. This entire procedure comprises of the preclinical part of vaccine development. The next part is testing on human beings, and it is known as clinical trials. And this part will be covered by my colleague Haroon Rashid. So I'm passing on the presentation to my colleague now.
Good evening. Is My colleague Aphrodite has explained what's a vaccine, what is the difference between a vaccine and a drug, and how the phase of development uh, is started like this preclinical phase. So I will explain the clinical phase of development of a vaccine. First of all, let me tell you before going through this graphical representation of vaccine development, let me tell you who are the key players in drug or vaccine development process. Number one, Number one is regulatory agency. This agency approves a vaccine or drug for human testing, as well as this agency gives marketing approval to a drug or vaccine. Uh, Here in US, we have FDA, US FDA, Food Drug and Administration. In India, we have Central Drug Standard Control Organization, CDSCO. And in Europe, we have European Medicine Agency. Number second, a principal investigator or a medical officer. Number third, are the scientists or clinical researchers. Number four, the important one, volunteers or the trial subjects or those participants that will participate in this study, in the study of vaccine or drug development. Number fifth, of course, the sponsor or the pharma company that will fund the process, this development process. This drug and vaccine development process is usually the same. The difference is we use drugs to treat the disease and vaccine to prevent the disease. And vaccines are biological products. So main difference, what is the main difference between a vaccine development process and a drug development process is that in drug development process, there are four phases. In each phase, in first phase, we enroll healthy volunteers, and in the remaining phases, we enroll patients. But in vaccine development process, in every phase, healthy volunteers are enrolled. So coming to this graphical representation, as you can see, after preclinical phase, we have phase one, phase two, phase third, and phase four. Briefly, in phase one, we do this phase one in small number of healthy volunteers, 20 to 100, and we assess the vaccine safety and immunity. In phase second, the number is increased from 100 to 20, from 20 to 100 to in hundreds. And in this, we assess the vaccine immunity, dose, and safety. And in phase third, what we do, we check the vaccine efficacy and safety. This is done in thousands of healthy volunteers, and it is usually multicentric. And after marketing of a vaccine, we do post-marketing surveillance, where this because every vaccine has some adverse effects, every drug or vaccine has some adverse effect. Most of these adverse effects we are not able to know in these phases of because these are limited and these are done in controlled environment. But after after marketing of the drug, we do surveillance of these drugs. We see whether there are any rare adverse events due to these vaccines or drugs. Coming back to the vaccine testing process, now I will explain the development cycle of a vaccine from base to bedside, that is from lab to clinic. Phase one. In phase one, these are the safety trials. In this, what we do after preclinical study is done, a IND, a file is submitted into a regulatory agency. Suppose in US we have FDA, Food Drug and Administration. Here we have CDSCO. We submit investigational new drug application to the regulatory authority, and that regulatory authority has 30 days to approve the application, whether they will approve or they will reject. If they IND application is approved, then this vaccine is subjected to human testing, phase 1, phase 2, and phase 3. In phase 1, vaccine is given to a small number of people, 20 to 80 healthy subjects. What we test in this phase 1, we test the safety and dosage, as well as we confirm whether it stimulates the immune system. Phase second. After phase, usually phase first is complete in 1 year. In phase second, vaccine is given to hundreds of people. And in this, children and elderly are also enrolled. And in this, we see if the vaccine acts differently in them. And these trials, in the phase second, these expanded trials further test the vaccine's safety and ability to stimulate the immune system. In phase third, these are the efficacy trials, and this is the main phase of the development, drug or vaccine development phase. It is done in thousands of people. In this, usually phase third tests are usually randomized, double blind, and it involves the experimental vaccine being tested against placebo. What randomized means, in this we allocate these patients or trial subjects in different groups. Double blind here means investigator, the medical officer, the principal investigator, as well as the patient or the healthy volunteer who is in their trial, they don't know what is given to them. They don't know about the investigational drug or vaccine. These trials determine whether the vaccine protects against the disease. For example, in this COVID, we will see whether this vaccine protects against the coronavirus. And in this trial, because these are done in thousands of patients or thousands of healthy volunteers, we see whether there are any rare side effects that we have missed in earlier phases of study. Then after phase third, we again, like after preclinical phase, we submitted a file IND, investigational new drug application, to regulatory authority so that they will approve for us to go for human testing. Similarly, after phase third, we have to again apply to regulatory authority, but this time it is usually in drugs, it is known as new drug application. In case of vaccines, it is known as biologic license application. In this, we submit this every data from phase 1 to phase third. We submit the data of efficacy, safety, immune response of the vaccine. We submit all the data to regulatory authority, and they will decide whether they will approve this vaccine or not. If they will approve, then this pharma company can market that vaccine. I want to tell you, if there is a pandemic or a public health emergency, this regulatory authority can give a uh, emergency use authorization before getting formal approval. Now, I will tell you briefly about this.
So, hi everyone. So, as my colleague explained very nicely about the clinical trial part of the vaccine development, like I had said right in the beginning of the presentation, COVID-19 was not really a part of the original presentation scope, but because right now we are in the middle of a pandemic, it is our responsibility as biomedical scientists to share a little bit of our knowledge with the wider world. So, in that aspect, my colleague is going to discuss about some of the um, he's going to discuss in brief about all the drugs, sorry, all the vaccine candidates that are in trial right now. And in particular, he will be focusing on one or two vaccine candidates that seem to be most promising at the moment. So, I will let Harun continue with his presentation.
Thanks, Aphrodite. First of all, let me briefly tell you about this COVID-19 or this pandemic. Is we have heard a number of definitions of virus, but the definition by Jean and Peter Medauer is appropriate. He says that the virus is simply a piece of dead news wrapped up in proteins. This RNA virus, a genome of 30,000 base pairs, it has brought continents, countries to stand still and delivered a major blow to our economies. This COVID-19 was first detected in Wuhan, China in December 2019, and thereafter it spread to whole world. And on March 11th, it was declared as pandemic. Till date, 49.7 million individuals have been affected worldwide, and nearly 1.25 million deaths are reported. In our country, as on 7th November 2020, it has affected around 8.4 million individuals, and more than 1 lakh deaths are being reported. A large number of people are becoming critically ill in short span of time. What they does, it overwhelms our hospitals, healthcare facilities. There is shortage of hospital beds, there is shortage of equipment, there is a shortage of manpower, healthcare, there is shortage of healthcare providers. And what we have at the moment, we are relying only on quarantine, isolation, infection control measures, so it prevents disease spread, and we provide supportive care for those who are infected. So the world is facing a major health catastrophe and economic devastation. One of the definite solutions is to have a safe and effective vaccine in this shortest possible time. But we must remember, no vaccine should be approved for use until safety and efficacy have proven. US FDA has drafted some guidelines for approval. According to them, the efficacy of a vaccine should be at least 50%, and the safety information should be from a large number of the study should have a large number of participants, around 3,000, and this should be for at least 6 months. The study should be conducted at least for 6 months, and there should be longer safety monitoring if any novel agent is used. There are also some strategies that how we can shorten this vaccine timeline, because usually a drug as well as a vaccine, it takes a number of years, 10 to 15 years to get approved. But during a public health emergency, some strategies are being followed so as we reduce this vaccine timeline. What we do, we test a number of vaccines simultaneously. For example, in case of COVID-19, 140 vaccine candidates have been have entered in preclinical or clinical phase. And what we do, we run different phases concurrently. For example, phase one, phase two, phase third, these are run parallelly for these vaccines and drugs. And sometimes these, if any vaccine is found efficacious or safe in phase first or second, then the regulatory authority gives the emergency approval. We scale up production while vaccines are tested, and challenge studies are done to test vaccine efficacy. These challenge studies, these are usually uh, there is a debate on going on whether we should do the challenge studies or not, because in this the participants are the healthy participants are exposed to a disease, to this virus, then vaccine is given. So there are a lot of issues whether we should, there are ethical issues whether we should do these challenge trials or not. Here is a graphical, well, you can see the race for a COVID-19 vaccine. If a vaccine, usually, if it will follow the usual phase preclinical timeline, it will take around matlab, 10 15 years. A vaccine will be in the market around in 2036. But we have shortened the timeline. We have a number of vaccines, like I said, 150 vaccine candidates have been simultaneously tested in preclinical and clinical phases. There is emergency approval also. And so the vaccine usually that is supposed to take 10 to 15 years will be approved in early 2021. The current status of these vaccines are in phase one we have this is the updated data. In phase one we have 36 vaccine candidates. In phase two we have 14. In phase three right now we have 11 vaccine candidates. And this limited, there are some uh, countries that have given limited or early approval. Without going to phase third, they have given the limited, or like China and Russia, they have given limited approval to their drug, but that has some serious consequences. So only 11 are in this phase three, and none of vaccine has being approved till now. In phase three trials, these are the uh, vaccines that are in phase three, and the phase three studies are ongoing or being planned. One is uh, by Moderna, one from AstraZeneca, one by Pfizer, one by Sinopharm, one by Sinovac, one by Casino Bio, one by Johnson and Johnson, one by Sanofi. Here I will explain two vaccines. One is that Oxford vaccine. It this in this vaccine, the study was also published in Lancet by Foligeti. It was a phase one, second single-blind randomized controlled trial. It was a multi-center trial done in 5 trial sites in UK, and the patient sample size was 1,077. And currently, it is in phase three, and in phase three, around more than 40,000 participants are participating in this study. It started in 28th August 2020, and it will complete, according to them, it will be completed in 25th October 2022. And as per the latest information, they will be distributing 100 million doses by second or third quarter of 2021. Another vaccine is Covaxin. It was developed by Bharat Biotech in a collaboration with ICMR. In phase one, there were 375 participants. It was done in 12 sites across the country, and it was completed in August 2020. In phase second, the study was planned first in 750 patients, but actually the study was done in 380 patients only. And in this month, they will release the results of this phase second study. And the phase third is the proposal is submitted in aims for ethical clearance. Maybe it will be done in, because phase third is done in thousands of patients, so it will be done around 26 to 28,000 of patients, and it will be done in 13 to 14 states. Now, Aphrodite will explain you briefly how to protect ourselves and others from this COVID-19 or coronavirus.
Thank you, Harun. Uh, so, since he is the expert on these things, he has very nicely summarized all the current ongoing trials for different vaccine candidates, etc. And as I have explained to you, so vaccines are not the magic components that are generally that they are generally projected as. There are lots of cons that are associated with them, but what vaccines do is that they give us a better fighting chance. They cannot guarantee complete protection from any pathogen, but the more number of weapons we have to fight against a pathogen, the better chance we have of protecting ourselves against pandemics of these kind. But when it comes to COVID-19, at this point, we do not have a vaccine. And even though experts in the particular field in our country as well as globally would like to have us believe we might have vaccines as early as February or March 2021. But I am not sure with my limited expertise how how much feasible or practical that is. So at the moment, we have got certain, even if they are not as effective, but they are weapons regardless. And some of them are being told to us repeatedly. Some of them are that first of all, you have to maintain, I don't like to call it social distancing, I like to call it physical distancing. So, first of all, maintain physical distancing. 6 feet is the limit that has been decided by experts in the field. Apart from that, what you need to do is that wear masks. It's not a particular problem in our country, but some countries seem to be struggling with the very idea of wearing a mask. Other than that, any kind of, like, you have all heard about the preventive measures that you can take. I will very briefly summarize why those things are actually effective. So what happens is that it's very difficult to, I mean, when you look up at the size of a coronavirus particle, imagine one particular, imagine the size of 1 millimeter and then divide it by 10,000. That's the maximum size of a particle that can carry that is carrying a coronavirus. Like, it is the known size of particles that are carrying coronavirus around at the moment. 1 millimeter divided by 10,000. So it's very difficult to actually take a molecule seriously, which is this small in size. We cannot see it, we do not know where it is, and over the period of time, people get very complacent about these things. But the thing is, as biologists, we get to understand, like, this is a very common occurrence in our lives as biologists. When we study immunology, a lot of us actually become paranoid because it seems like there are millions and millions of small particles which you cannot see with your eyes but which can actually kill you. And worse than killing, it can actually cause very bad damages to your body, and you will be alive but you will have lost a lot of functions in your body, which is the case that is happening in case of COVID-19 infection as well. As it is, COVID-19 symptoms seem to be resembling that of common flu. But then the a large fraction of the patients who are getting infected with COVID-19 are not really expressing very intense symptoms. But what happens is that a large fraction of those patients are experiencing long lasting effects. Among them, the most serious are multiple organ damages. They are causing permanent damage. So far, what we know is that they're causing permanent damage to your lungs. They are even causing, like, an everlasting sense of fatigue. They are causing neurological issues. Most of them are centered around attention, memory, and concentration, etc. So the thing is that we do not know a lot about what this virus does, but we cannot see it. That does not mean that the virus has stopped infecting us. And at the moment, we do not have any proper drugs to treat the virus, and we are still waiting for a vaccine. So until we have some definite weapon in our hand, what happens is that the lesser you expose yourself to the virus, the more safe you are. And it is in line with that logic that you are asked to maintain distance from most people around you. You are asked to follow basic hygiene guidelines, which is washing your hands over and over again. You are asked to wear a mask because every time you talk, you release droplets out of your mouth. Every time you sneeze, you release droplets out of your nose, and it can infect other people. A mask that you wear will most probably not protect you against the virus, but it will protect others against virus that you may be releasing. And in the same way, when others are wearing masks, then you are protected against the virus. Nobody can guarantee 100% protection, but these little things are weapons that we have at the moment to fight against the virus. And as people, you people who are attending this talk right now, I don't know how many of you have biological background, but I would implore you to take this seriously, to make people around you understand the threat that we are facing. Even if we do not show symptoms, we infect others. We do not know who gets to die and who gets to live. We do not know what long-term consequences we are faced with once infected with the virus. So it's better not to take any chance. And apart from that, everybody should have trust in science. And there's nothing else to say. The conclusion of this presentation is that, of course, vaccines are our best hope right now, but we should also hope that they go through stringent protocols and all protocols are followed properly and all risks are measured properly so that the public at large is not faced with even graver challenges. Like on an ending note, I would like to not exactly quote, but there are concerns by a group in San Francisco. There's a professor, I forget his name, but they have a very like they have a very valid concern. They are a group that researches on HIV, and HIV vaccines that they have been studying for over 20 years are based on adenovirus vectors. Adenovirus vectors are the same vectors that are being used by the Oxford group, and which is our best vaccine candidate at the moment. And that particular adenovirus, which is adenovirus type 5, is has been considered a candidate vector for HIV vaccine for over 20 years before it was observed that people who were vaccinated using those vectors were actually displaying signs of increased vulnerability to HIV infection. So the professor and his group has actually expressed concerns that it might be possible that even in case of COVID-19 vaccination, the adenoviral vector might increase the risk of vulnerability to the disease in people who get vaccinated with that particular vaccine rather than helping us. So, in summary, because we are working with biological agents, predicting everything to the point is very difficult, and it might take years and years of study to actually determine what causes what and how something is affecting something else. At the moment, everybody should just trust science and follow what the experts say. That's it. Thank you very much.
Thank you, Aphrodite. It was such a nice talk. It was a very clear idea that you people presented. Also, thanks to Harun. And I think now we can start the discussion sessions. If anyone from the audience have any question, you may ask now. Or if anyone wants to give any comments about
Yes,
both the talks.
I do have a few questions.
Yes, please say your name, and then you ask the question.
Well, first of all, thanks to the speakers for such nice elaborations. I'm asking this the first question from a purely non-medical ground, and the question is that what is the basic difference between hard immunity and adaptive immunity that you are talking about?
Okay. So, Kinkar, is that your name?
Yes.
Yeah, so Kinkar, very nice question. Again, in the present context, it is actually a very, very good question. The difference between adaptive immunity and herd immunity is that the latter depends on other people. To explain it better, adaptive immunity is a immune system, is an immune response that your body keeps on building up over the years. Herd immunity is something which basically involves the contribution of, let's say, you are living in a particular society. You are 20 people. One person, each of those 20 people will have their own adaptive immune system. But let's say a novel virus comes in. That virus will have that the body, neither of your bodies actually know that that virus exists. So you are all 20 of you are getting exposed to that virus for the first time. All let's say 5 of you get infected. In the first instance, 5 of you get infected. Okay. All 5 of you will start either showing symptoms or some kind of an immune response. But let's say over an extended period of time, 8 of you have already been exposed to that virus already. 8, or let's say 10 of you have already been exposed to that virus already. Your body recognizes those viruses. Now, let's say you're meeting with another person from your society. That virus is trying. We are talking about a second infection. Okay, the other person that you're meeting is actually carrying the virus for the second time. It is trying to jump onto you, but you already have immune system responses to it. So that when the virus comes inside your body, it just dies. Same way, when somebody else from your society is going and meeting that person A who was infected for the second time with that virus, that person, the other person also has immunity to it, so the virus again dies. In this way, the virus has lesser people to jump onto. It goes onto the other people and it just dies because your body already knows that the virus exists. So when in a larger population, let's say in a population of 1,000 people, at least 75% of the people have already been exposed to the virus for the second time has got much less people it can jump to in whom it can survive. So the uninfected people have got protection because the chances of survival of the virus decreases. That is what herd immunity is.
Okay.
Yeah.
Okay. So maybe I'm missing something on or not. I don't know. But uh, what then could be the use of vaccine when herd immunity is already developed?
So first of all, herd immunity is not 100% effective. Okay, because your body cells, as it is happening, as it is being reported in the case of COVID-19 pandemic, people seem to be losing their immune memory. They seem to be getting reinfected.
Okay.
Okay. So in
Okay.
that case,
Okay.
herd immunity is not at all going to work. If let's say if only for 3 or 4 months your body remembers that virus, then herd immunity will never be developed. Okay, your body has to
Okay.
remember it for a long period of time. Like even after 1 year, if the virus starts coming back again, your body has to remember it in order for herd immunity to be there. So in cases like
Okay.
that, vaccines like I'm not sure you know about flu vaccines. But what happens in US is that they have to take it every year because the the response of the body gets forgotten. Okay, so every year you have to refresh its
Right.
memory. So in that case, herd immunity is not at all possible because it's not possible that in 1 year about 60 to 70% of the people get infected every year. Okay, so in that
All
case,
right.
you need a established vaccine that every year you can take the shot and you can get immunized all over again.
All right. Okay. Okay. I got it. I got it. Thank you so much. And then you talked about during the course of vaccine development, you have the as a first step a simulation or some use of some softwares.
Yeah.
Right. So I'm just curious to know about how does this simulation work? I mean, can you please give a brief
So there are
description?
very highly priced softwares, okay, which basically do modeling predictions. Okay, I am not exactly an in silico expert. I have never done that work myself. I am more of a molecular biologist. I do the tampering with the cells. So, but from what I have learned about it in my research experience is that they have got very expensive softwares. Like in biology, there is this software called Schrodinger, which is priced at over 8 lakhs and 8 lakhs or 8 crores. I don't really remember, but anyhow, that predicts the protein modeling. Okay, so it basically predicts that let's say you know that a particular protein behaves in a certain way and it interacts with other receptors in a certain way. But before investing in a lot of chemicals and reagents and lab time, what you want to do is that see in the software whether or not they're binding together, whether or not their sites are compatible with each other, whether or not they have proper like 100% binding or whether it's like something else is coming and binding in its place. Okay, some other like in the cell there are many components, whether or not some indigenous component of the cell is coming and binding in a place and this molecule of interest is not at all being able to bind. Okay, so those binding dynamics, those binding efficacy is checked through software before you invest in lab.
Okay. Okay. But say for example, as we can see that for those patients having some comorbidities of any kind, the COVID-19 virus is more effective or more dangerous in case of them. So, in case of producing or developing the vaccine for that, in those simulations for some particular viruses, for some particular vaccines in against some viruses, so in those simulations you should have some particular conditions or constraints that have to be implemented, right? So
Yes,
what are
yes.
the conditions that you choose? I mean, for example, in case of coronavirus, the comorbidities like whether the patient is diabetic or not, whether the patient is having high blood pressure or any heart issues, etc. What are the conditions? There could be a numerous number of conditions or constraints that you have to put in your modeling. So how do you do that? I mean, how do you choose your
So
conditions? most effectively?
Practically speaking, since I've again never used it myself, I wouldn't be completely sure. But going by
Okay.
established practice, what you do is that you establish a panel of molecules that are already in, let's say, in a diabetic patient. Some kind of molecules are dysregulated. Okay, something is extra, something is less. Okay, so the bad things actually become extra in case of any disease. So let's say, in the most layman terms, in a diabetic patient, a particular kind of molecule is present in extra amounts. Okay, so
Mhm.
while doing the simulations, you make a panel of all those molecules which are dysregulated. If you would like to study disease specific
Mhm.
response to particular vaccine, but I don't know if that is done. I think the more practical approach is that you take volunteers who have different kinds of conditions, and during the clinical trials, you just administer the vaccine and you see, because once it is in the clinical trials, it is
Mhm.
not expected to cause very adverse effects. Sometimes there are very adverse effects, but particular disease related correlations are done more in the clinical trials rather than during the preclinical trials. Okay, but if you had a lot of time and if you had a lot of curiosity, I'm pretty sure that computer based biological workers do this all the time. They create panels of molecules. Okay, and then they see what
Okay.
happens when there's a molecule of your interest. They see what happens, what is the kind of interaction that happens with that panel of molecules. So let's say there is a bad guy out there, a bad molecule out there. Your vaccine candidate is interacting very strongly with that bad guy molecule. It is stabilizing the bad molecule. So you don't want to use that vaccine candidate because it is pretty sure that when it is stabilizing the bad molecule, it is going to cause a bad effect on actual human beings. So that's how it is.
Okay.
It would be done, but I'm not really sure that that is done in silico process, because that is more of a domain where you study in cellular environments, or where you study in animal models. You know, animal models are created for this thing. So, like, you create diabetic models, you create neurologically disabled rodent models, you create other animal models, and you test whether the vaccine is doing something bad to them.
Right.
Okay.
Okay.
Yeah.
Okay. Thank you so much, Aphrodite.
Yeah.
Thank you so much. Yep.
Okay, so we have one question in the chat box by Deeptak Bishwas. He says, I have a general question. Why the second or third wave of this COVID 19 infection is looking dangerous than the previous one?
Okay, so I will let Haroon answer that question because I honestly don't have much knowledge about epidemiology.
Okay,
The question is about the second and third wave. Hello.
yes. The question is why the second or third wave of this COVID 19 infection is looking dangerous than the previous one.
Actually, if we will, it is not the first wave, second wave, third wave. There is no such difference that first wave was not that dangerous than the second wave or the third wave. It is actually we know the problem now, and we it first when the first wave was there, we was not knowing about the problem. So, but I don't think the second wave or the third wave is more dangerous than the first wave.
Okay, I hope
and
Deeptak
and
has.
and and this time only thing is that because due to the seasonal conditions, for example, like in India we have winters here, the second wave or the third wave, maybe this in India it is a second wave. It may be dangerous because winter is coming. There is we call that double pandemic because we have seasonal influenza as well, we have COVID-19. These both things will together will cause a increase the problem. It will overwhelm our hospitals, our because uh, intensity and of the virus is same, but since our hospitals and the manpower is already overwhelmed, the second wave or the third wave will create more problems. For example, we have hospital with 100 beds and already 90 beds are occupied, and second wave is coming. there we don't have any more beds for those patients, and we don't have manpower for that, and we don't have, and till date we don't have any drug or vaccine to treat them. So second wave or third wave in that sense is more dangerous. And in India, in our country, winter is coming, so this may cause more problems. More patients will be prone to this COVID-19 infection.
Hello, Haroon. Can you hear me?
Yeah, yeah,
Yeah. So what I am basically asking that, okay, so if you just look into Europe or America, so the number of infection is basically increasing. I'm just asking
yeah.
out that why? Because now we know that.
Actually, this is called COVID-19 fatigue. Because
Okay,
now now we common people, after so many months of lockdown, so many months from so many months we are wearing masks, doing this hand sanitization. People are tired of it. They some have removed their masks, some are going to restaurants, some are moving out. So this is increasing this number.
okay. So
So
this
that is
makes.
why in many in Europe they are again locking down their cities. This is actually known as COVID-19 fatigue, because people are tired of these masks and everything staying in their homes. They are moving out now. So moving out increases their infection.
Yeah, so I would also like to add to this. Like speaking from a molecular point of view, viruses mutate very quickly. Okay, so at the moment, such kind of mutations have not happened yet. Okay, like right now there are like six strains of virus going around, and more like all of them are general. Like whatever we have been seeing in the first wave, that is what has been continuing till date. Okay, and the problem is that historically speaking, in the like we have been quoted the 1918 Spanish flu quite regularly. Uh, in that case, as far as I know, the problem occurred because of a highly virulent mutated strain of the same virus. Like, of course, as my colleague said, there has been a what was that fatigue, COVID-19 fatigue thing. Okay, that is what I was also saying about that people have stopped taking the thing seriously. Okay, in the beginning, we were also paranoid, we were also terrified, we were staying in our homes, and we were following directions very stringently. But as time passes by, people tend to ease up on their caution. Okay, that is of course a very huge sociological factor which contributes towards a higher number of infections during second, third, fourth, whatever number of waves. But yeah, from a molecular point of view, there's always a huge risk that a virus mutates because viruses mutate very quickly.
Okay. So thank you, Deeptak, for asking your question. Anyone else having any
Yeah,
other
Shreya,
questions?
I have a question. Sorry for.
Yes, sir. Yes, sir. You may
Yeah.
ask.
So thanks to the speakers for the nice talks. So I have a question. Means like our children, they are going through normal vaccination. Okay. So is the COVID-19 risk is somehow less for them, like 2 years old baby or something like that?
So, sir, sir, thank you for the question.
Yeah.
But so far, studies about risk to children have not been conducted on that kind of scale, whether they are immunized because of the vaccinations. But what has been said is that children have less amount of ACE2 receptors. Pretty sure you've heard about the name. To explain to everybody else, is let's talk about the analogy of a lock and key. Okay, viruses have a key that unlocks a particular lock in our human cells. So ACE2
Okay.
is the lock, and this COVID-19 virus has got a particular key in its spike protein. Okay, and it is using that key to put it into the lock that belongs to our cells, which is the ACE2 receptors, and it is entering the cell and it is replicating in large numbers and infecting us. But kids have
Okay.
a lesser amount of ACE2 receptors, less locks, less infection.
Okay. That's why the risk is little bit less for children.
Yes, yes, they get
And
infected, but the virus doesn't stay alive in them, in most of them.
Okay, okay, okay. Thanks a lot.
and
Thanks a
I
lot.
will add to this.
Yeah.
Usually, these children are being given this influenza booster vaccine,
Yes, yes.
and they decrease the chances of COVID-19 infection.
Okay.
And that
Okay.
is why some of the experts are saying to because now we have this winter is coming.
Huh?
Some of the experts are of the view that if we will get influenza booster vaccine, both
Huh?
children and adults,
Okay.
then the chances of COVID-19 infection or the severity will be less.
Okay. Yeah, thanks a lot for the children part. And
Okay.
you just told that these two vaccines can be taken by the adult as well. So is it possible
Yeah. By adults as well.
at this time we can go also for this vaccine. Like
Yeah.
so we can
The,
just
the, these these influenza booster vaccines are safe as well as available. We
uh,
can have them, and it will decrease the severity, and
okay.
it will decrease the chances to get this COVID-19 infection.
Okay, so for this we can just consult a doctor and ask them to for this vaccine, right?
Influenza booster vaccine.
Influenza booster. Okay, 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 okay. Thanks a lot. Thanks a lot.
Okay.
Yes, I was just going to ask about a similar question that Shoykant has asked. So while developing a vaccine, do you keep in mind the age of a person? I mean, is there any age constraint that a vaccine should be applied to a person of certain age or not? Is that a constraint in course of development of it?
No, it is not like that. That is why in this phase third, we are phase second, we test this vaccine in children as well as adults, so that when it will go to general population, it will be administered to everyone, irrespective of
Okay.
age.
Okay. Okay.
Because
Thank you. So,
COVID-19 virus is affecting everyone, children as well as adults as well as elderly. So
uh,
this vaccine is for everyone, irrespective of age,
okay.
gender.
Okay. Whatever be the risk factors are depending on the ages.
Actually, in phase third, this everything is because we give this vaccine to every population. Those with
Okay.
risk factors, those with comorbidities, for example, with diabetes, with
Hmm.
hypertension, and pregnant
Hmm.
women as well. And
Okay.
we see whether there is any serious risk with that vaccine or not. For example, if a
Okay.
vaccine has a serious risk in case of a pregnant woman, then we
Uh-huh.
contraindicate it that this vaccine should be given to this population, but not to
Okay.
pregnant women. So
Okay.
these
Okay.
those vaccines are contraindicated. But in some cases, after marketing the vaccine or drug, we found that after marketing, after 1 or 2 years, we find that this vaccine causes problems in this population. For example, elderly population, or children or pregnant population. Then we change the labels of the drug. We change the labels like this drug will not be given to this pregnant people or these people with diabetes, or if it is causing problem to general population, then we ban the drug. We black box the drug.
Okay. Okay. Thank you so much, Arun. Thank you so much.
So, are there any other question? Maybe is there any question from the Facebook or YouTube? I can see there is no. Hello, is there any other question?
Yes, there is one in the
Okay,
chat box.
so yes. Uh, so it's Oindrila Chakraborty. The question is influenza vaccine is safe for hyponatremia, hypokalemia patients? Hello, Aphrodite or Harun, can
Hello,
you hear me? Yes.
can you repeat the
Yes, the
question?
question is written in the chat box
Hello.
actually. The question is influenza vaccine is safe for hyponatremia, hypokalemia patients?
Hello.
Yes, hello.
Uh, I'm
Uh,
saying that usually this vaccine is safe for even those population, even though it causes hyponatremia. This vaccine, but it is safe.
okay. I hope you got the answer, Indrila. If there are no other questions, I think it is time that we close the event for today. So once again, thank you to both the speakers. The presentation was really nice and beautiful. I hope we will all in the future look back to this presentation once again and the YouTube link. Thank you
Thank you,
very much.
Shreya. Thank you to the CAPS group for giving us the opportunity.
And also thank you to the audience for patiently listening to the talks. So let us hope to meet you once again next week. Thank you, everyone. We may close the event now. Bye.